Good morning, everybody. My name is Micaela Grandolfo. I work at SISSA, the International School for Advanced Studies here in Trieste. And my talk is about optimization of specimens for fluorescence microscopy. I have to confess, I was really in trouble when Alessandro told me you can speak about optimization of specimens, because as you for sure know, there are a lot of informations or advices that I or everybody working in this field can give you about how to optimize a specimen for fluorescence microscopy. So I decided to cover just a few topics, because this is just a very brief overview. We do not have a lot of time. That are very important, in my opinion, due to my experience in the lab with students, PhD students, undergraduate students, and so on. And something regarding the fixed sample and some for live sample. So, when you are working with fluorescent microscopy, you see something, and then, of course, you want to report what you see. It's true. But are you every time really sure that what you are seeing is true, is biologically and physiologically true? Because this point is very important. If you start to design your experiment with this question in mind, you can avoid a lot of artifacts and you can work unbiased. Okay, so this is a very important point. Okay, so when you work and you want to acquire images in fluorescent microscopy, you have to go through 3 critical steps: the sample preparation, the image acquisition, and the image processing. I will stay on sample preparation, of course. Okay, I wanted to speak about 2 fundamental rules. The first one in sample preparation is, remember that garbage in means garbage out. What I would say is, if your sample is not a good sample, if your staining is a bad staining, if your fluorophore is not the proper fluorophore, if your antibody is not recognizing properly your antigen, forget to have a good image just because you have a fantastic new confocal microscope at the 2nd floor of your institute, okay? You have to spend some time and try to obtain the best sample that you can, okay? So spend some time to think about it and to design properly your staining experiment before going to the microscope. Okay, the second rule is in general this one. So it happens very often that in every lab there is just one standardized protocol to do immunofluorescence. And this is totally wrong, because of course for every sample you have to think about it and you have to choose the proper fixation, the proper permeabilization. You have to think about the antibody you want to use, the fluorophore you have to use, and you have to collect as much information as possible to perform a good staining. So this is not true, and what you have to obtain is a dedicated protocol for each experiment, for every single experiment. Okay, so fixed sample, in particular for immunofluorescence. So for every sample, you have to optimize these passages: the fixation, the permeabilization, the blocking, the choice of fluorophores, and the mountant. We will skip the permeabilization and the blocking, and we will move to the fixation immediately. I want to speak about fixation, just taking into account the problem that you can encounter using the 2 main type of fixation. I mean the problem of autofluorescence, the fluorescence due in this case to fixative. Okay, you can use cross-linking fixative like formaldehyde or glutaraldehyde, or you can use reagents that precipitate proteins like alcohol or acetone. So, fixative induced fluorescence. Remember that this type of autofluorescence is due to reaction of aldehydes groups with tissue components, and it is worse with glutaraldehyde and when the fixation is warmer or longer. If you can observe your tissues or your sample using a long pass filter in the green region, you will see something that is not bright green, but it is a dull yellow. In this case, you can have the suspect that this is autofluorescence. When it is very homogeneous, you do not see some specific staining. But remember that you can have different intensity in different cells due to different contents in terms of amines, okay? Okay, in general, you will use formaldehyde because it is easier to work with formaldehyde. And remember that the first tip you have to have in mind is to prepare freshly solution. If you prepare fresh solution from paraformaldehyde, you will have small polymers in solution, and small polymers are able to fix in a better way and faster your sample, and you will have less autofluorescence in your tissue. Okay, then remember that autofluorescence in mammalian cells and tissue is reduced in longer wavelengths. Okay, so like in this example, you can see that here this tissue was stained with two different antibodies, one in the green range of the spectrum and one with the fluorophore for the red range of the spectrum. As you can see, in the green range, okay, this antibody doesn't work properly, but this is another problem, but you can see that there is a lot of autofluorescence, okay? And you can distinguish, it is not a good staining in the red region of the spectrum, but you can distinguish the staining in this case. So, another trick you can take in mind is if you have problem of autofluorescence, you can decide to use red emitting fluorophore, okay, just to avoid to work in the green region of the spectrum in which the autofluorescence is more severe. Okay, so remember, the redder the better in this case. Okay, alcohol and acetone fixation. In this case, you will not have problem in terms of autofluorescence, but be very, very careful because these reagents denaturates proteins. So if you have fluorescent protein like GFP, YFP, M-cherries and so on in your tissue, if you fix your tissue with alcohol or acetone, then it is very hard to look at the fluorescence of your proteins. So be careful. On the contrary, this type of fixation could be very useful if you have Lipofuscin in your sample. Lipofuscin is a breakdown product of red blood cells. You can find Lipofuscin in muscle cells, cardiac cells, glial cells, neurons, especially in old animals, for example. And you can, if it is possible, concerning your protocol, to fix your tissue with alcohol and acetone, you will produce an extraction of lipids. This way, you solve the problem of Lipofuscin. Okay, you remove it from your tissue. Okay, so besides of Lipofuscin, you can find also elastin, that is a fluorescent molecule in the tissue, or collagen. So there are some protocols to quench the fluorescence of Lipofuscin and elastin. I don't want to go into detail in this protocol. I just want to underline that you can find this fluorescent molecule in your tissue, and it is important to recognize them and to know that you are not looking at your antigen, but there are some fluorescent molecule present in the tissue. You can find also an ADH or FAD. In this case, an ADH emits in the green-blue region of the spectrum, and FAD in the yellow-red region of the spectrum. Okay? Okay, so methods to counter autofluorescence are freshly prepared PFA, as I told you, a treatment with glycine, so you can feed the free aldehyde groups with some amino groups, in this case with glycine. You can use borohydrate to reduce the aldehyde groups. And in the case of Lipofuscin and elastin, you can quench the fluorescence with Sudan black or toluidine. You can search in the net. There are a lot of protocols concerning the use of Sudan black or toluidine blue. And finally, you can extract the Lipofuscin with alcohol or ethanol or acetone fixation. Okay. Another method to counter autofluorescence is to play with acquisition. It is a little bit dangerous. You have to know exactly, perfectly what you're doing. So be very careful when you play with acquisition. But for example, this is just an example. In this case, this is a section of vomeronasal organ of a mouse, and what we were interested in were just the cilia of these apical cells. So the antigen was located here in the cilia, and the other light coming from the other part of the sample was just autofluorescence. So you can play with the acquisition to obtain the staining of the cilia alone. Okay. So, I would like to underline that it is really important to check for autofluorescence in your sample before going ahead with your experiment and before reaching some conclusions. This is an example. It is not very clear, but okay. This is a section of brainstem, and this section was stained for a nuclear protein. Of course, the tissue was fixed and incubated with primary and secondary antibody. The secondary antibody was conjugated with Alexa 488. Okay, so the first control was to remove the primary antibody and to incubate a fixed section just with the secondary antibody. And as you can see, the image is not very different from the previous one. The last control was to have a look of the section just fixed. And as you can appreciate, there is no difference also in this case. So my advice is do not stop here and try to find some conclusions. Every time you have to control for your secondary antibody and even for your, the eventual autofluorescence that you can find just in your fixed tissue. Okay? Okay. So let's move to the fluorophore. How to choose a fluorophore? So is it simply a matter of color? In general, people choose fluorophore because likes green more than red or blue more than green. No, it is not so simple. It is not so simple. Here we can summarize the characteristics of a fluorophore that Gabriele showed you this morning. You can look for the excitation emission spectra, for the quantum yield or the quantum efficiency of the fluorophore, the lifetime, the quenching, and the photostability or the sensitivity to the photobleaching. Okay, the excitation emission spectra. You have seen 10 of these diagrams this morning. So you have to know which is the excitation and the emission spectrum of the fluorophore you want to use, because this is the identity card of your fluorophore. And as Gabriele told you, you have to be very careful when you want to perform multiple staining to avoid overlapping of the emission spectra of the two fluorophores you want to use together in your sample. The quantum yield. So the quantum yield is a very important characteristic of every fluorophore. Is a quantitative measure of the fluorescent emission efficiency. And it is, in synthesis, the number of photons emitted per photon absorbed. So this number ranges between, between 0.05 and 1. So the maximum is 1 photon emitted per 1 photon absorbed. It is strongly environment dependent and it is proportional to lifetime. Remember that as you have already know from this morning, the lifetime is the duration of the excited state of a fluorophore before it turns into its ground state, and the scale is nanosecond. Okay. This is a table in which you can compare some quantum yields or quantum efficiency of different fluorophores. As you can see, there are fluorophores that have a very, very high quantum efficiency, like Alexa 488, very close to 1, or fluorescein, or rhodamine, can you see, marine blue. And there are some other fluorophores that have a very, very low quantum efficiency. For example, Alexa 555, 0.1, or Psi3. Okay? I would like to underline that the quantum efficiency is directly connected to the lifetime. So a high quantum efficiency means that the fluorophore has a high lifetime, a long, in terms of nanoseconds, lifetime. But what I want to show you is this particular fluorophore, the Alexa Fluor 633, for which the quantum efficiency was not determined. So it is so low that it is impossible to give a precise number to the quantum efficiency of this fluorophore. So this is a particular fluorophore because very often researchers buy this fluorophore because it matches perfectly with the 633 laser lines in the confocal configuration when you want to use with far red fluorophore. Okay. This is correct from the point of view in terms of efficiency of excitation because, okay, this is the 633 laser line, and this in blue is the excitation curves of Alexa 633. As you can see, you have the maximum of excitation using the 633 laser lines. But what it is better to buy, it is not Alexa 633, but Alexa 647. Because Alexa 647, this is the excitation curves of Alexa 647, as you can see. The efficiency in terms of excitation is around 55%, but, the quantum efficiency of this fluorophore is determined, and it is not the best that you can have, but it is something that you can work with, okay? So my advice is when you have to decide which fluorophore to use, do not stay just, do not fix to the quantum efficiency or to the efficiency in terms of excitation regarding the laser lines that you can use on your confocal microscope, but try to collect as much information as possible to decide which is the best fluorophore for your purpose. Okay, the quenching. So the quenching is an irreversible decomposition of your fluorophores due to interaction with a quencher, in particular, in general, it is oxygen. or with aliphatic or aromatic amines. As you can see from this diagram, of course, the emission is really reduced after the quenching of a fluorophore. And from this very, very simple cartoon, you can see that a fluorophore is excited by light. It goes to an excited state, and then it goes back to the ground floor emitting light. Or in presence of a quencher, there is this collisional quenching that moves the fluorophore to the ground state without emission of light. Okay, you cannot avoid this phenomenon. You can just try to reduce it using anti-fading molecules in your mounting medium. The photobleaching. Okay, the photobleaching is a covalent modification. It occurs when a fluorophore is permanently loses the ability to fluoresce due to photon-induced chemical damage. So also in this case, it is unavoidable because, of course, you need to excite your fluorophore with photons. But also in this case, it is really, really critical to choose the right fluorophore for your purpose, because there are fluorophores that bleach very, very quickly after few cycles of excitation, while others are more resistant, more robust, and can undergo thousands or even millions of cycles before bleaching. It is not easy to find this information in the net. Here there is a comparison between two couples of green and red fluorophore, Alexa 488 plus Alexa 546 and fluorescent inside 3, same sample and same type of illumination, and it is clear that the first couple is more resistant to photobleaching. Okay. What I would like to show you is this diagram. It was difficult to find this diagram, and it was impossible to find something like that for other fluorophore. This is a comparison between 7 different green emitting fluorophore. Can you see Alexa 488, Daylight 488, Chromeo 488, Northern Lights, and so on. It was used the same sample, the same protocol, the same type of continuous illumination, just to compare the resistance to photobleaching. It is clear that there is a different starting point, and this corresponds to the quantum efficiency of the different fluorophores. And it is also clear that some fluorophores are more robust than others. In this case, after 20 seconds of illumination, you can collect some light. In the case of these two fluorophores, you are at 0 emission in terms of fluorophore or photons. So when you have to buy, in this case, a green emitting fluorophore, of course you have to think about the price of the fluorophore. But remember that sometimes you do not need to have a very cheap fluorophore, but you need to have a fluorophore with a high quantum efficiency and very resistant to photobleaching. So do not compare only the prices, okay? Okay, method to counter photobleaching. Of course, you have to try to scan or to illuminate your sample for shorter times. You have to use superior quality optical components, I mean objectives with very high numerical aperture if possible. You have to use state-of-the-art detectors, as Giacomo told us a few minutes ago, to have very sensitive devices to collect as much fluorescence as possible. You have to reduce the excitation intensity using some neutral filter or reducing the power of the laser you are working with. You can use anti-fade reagent, and of course you have to use fluorophore, more robust. Okay, so I hope I have convinced you that not all fluorophores are the same, and it is really useful to spend some time to decide which fluorophore you want to use for your particular experiment, and not just use what you have in your fridge, okay, in the lab fridge. Okay, so the mountant, of course you have to use the mountant to preserve your sample. It is really important because it raises the refractive index of your sample. In this way, the refractive index will match perfectly the refractive index of the oil you will use for oil immersion objective. And in general, it contains scavengers to remove free radicals to reduce the quenching phenomenon. Okay. So, when you have to decide which fluorophore to use, remember they have different quantum yields, different lifetime, different photostability. Remember that human eyes are more sensitive to green, so if you want to show your result and you want to obtain a very strong impact on your audience, use green, okay? No, it's true, it's not a joke. Remember that autofluorescence is weaker at higher wavelengths, so play with different fluorophores if you cannot remove the autofluorescence in your sample. And if you have to do a multiple staining, use this is very important, use the best fluorophore for the most critical antigen, okay? Because you have to be really sure that you are looking at your antigen when it is really difficult to visualize, to reveal, and you want to use your best fluorophore just for that antigen, okay? Be careful with spectra overlap when you have to perform multiple staining. So, in synthesis, collect as much information as possible, okay, before starting your the staining with fluorophore of your sample. Okay. Okay, and now just few words about live cell microscopy. Also in this case, as you know, there is an enormous amount of useful information to collect, to try to design a good experiment in live cell microscopy. So of course we do not have time to cover this topic. You can find a lot of very nice, very good manuals and books on it. And I decided to concentrate on one single aspect of this type of experiment. The health of cells and tissue under investigation, just this. Because remember that in live cell imaging, every decision in the process you are building up impacts the final results. So it is very, very crucial how you take care of the health of your tissues and cells. So I'll skip these long lists of fluorescent proteins and dyes, and I want to start taking into account the conditions under which cells are maintained on the microscope stage, because very often these conditions dictate the success or the failure of your experiment. Okay, so to successfully image cellular processes in living cells, the cells must be kept in an environment that does not induce stress responses. I mean, you have to take care and you have to try to control as much as possible the medium, the temperature, the osmolarity, and the phototoxicity. Okay, the medium. In live cell imaging, you can use media very, very similar in terms of components of the medium that you use to grow your cell in the cell incubator. The point is that in our cell incubator, we have a buffer system that is based on bicarbonate, and it works in a controlled atmosphere in which there is a small percentage of CO2 or carbon dioxide, in general 5%. And it is tricky and expensive to reproduce this system around a microscope. So it is really useful to use some biological buffer like HIPPIs. But the point is that you have to be very careful, because HIPPIs is not every time the solution, because there are a lot of cell lines that do not tolerate a lack in carbon dioxide. So you have to check your cell line for this characteristic. Otherwise, moving to a HIPPIs buffer medium, you can change the physiology of your sample. Okay. So remember, each cell line should be carefully tested for the tolerance of a lack in carbon dioxide. Okay, the temperature. In live cell imaging, you have to control also the temperature. In this case, you need to work with large boxes that enclose the whole microscope, or small chambers. The advantage to work with large boxes is that when you reach the selected temperature, the system is really, really stable. The weak point is that it takes several hours to reach the temperature, and it is very expensive. In the case of the small chambers, you reach the temperature very fast, but you have to be you have to pay attention to a problem that it is very often underestimated, and it is the problem connected to the use of immersion objective. Because when you are working with immersion objective, an immersion objective function as a heat sink at the level, in the point in which there is the contact between the frontal lens of the objective and the field of view of your chamber. So in this case, you suppose to have a controlled temperature, but this is not true, okay? And what you have to do is to use an objective heater to be sure that the temperature you want to use is homogeneous all over your sample. Okay, osmolarity. So in general, we are in live cell imaging, we are using small volumes, and these small volumes during time can change the osmolarity due to evaporation that is more severe when you are heating your sample at 37 degrees, for example. So to avoid this problem, you have to work with sealed chamber, or if you're working with large boxes, you need to humidify the air over the sample. This is just an example of some hippocampal neurons. This is the starting point. Very simple phase contrast images, of course. So you can switch. Remember that every time you have a doubt, you can switch to the bright field configuration and have a look of your cells using phase contrast or DIC. It depends on which type of microscope you are using. This is the starting point. After a few minutes, upon evaporation, you can see there are some blabbing at the neural processes of the cells. Can you see? It is pretty clear. And after a longer time, the damage is more severe and the cells are detaching from the bottom of the chamber. So sometimes it is not clear when you are looking at your fluorescent molecule that something very, very dramatic is happening in your sample. So if you have doubts, switch to bright field to check your sample. Okay, phototoxicity. So the phototoxicity is an oxygen-dependent reaction due to free radical species. And these free radical species are generated during the excitation of your fluorescent molecules or dyes. So you cannot avoid this problem because, of course, you need to use fluorescent molecules and you need to have oxygen present in your sample. What you can do, you can try to limit this phenomenon, supplementing the medium with scavengers, such as ascorbate, vitamin C, vitamin E, and sometimes people, if there is the possibility to work in a controlled atmosphere, try to reduce as much as possible the percentage of the oxygen. Okay. So, to fight the phototoxicity, you can try to reduce as much as possible the amount of excitation light, also in this case using filters, reducing the power of the lasers, programming in a good way the shutters, the electronic shutters, for example, to avoid illumination when you are not acquiring imaging. Of course, you have also in this case to use detectors, the state of the art of the detectors to detect as much fluorescence as possible. Remember, also in this case, it is crucial to work with a very high numerical aperture objective. And you have to reduce the concentration of fluorescent probes to the minimum that is useful to obtain your result. Except of the phototoxicity, do not forget that UV light is very dangerous for the cells because damages DNA and RNA. And also focused infrared light is dangerous because it causes heating in a very small local area of your sample. Okay, so as I already suggested, it is very important to monitor your cells during the experiment. Have a look of the cell morphology using the transmitted light microscopy, DIC or phase contrast. Check for clustering of fluorescent proteins, that is an index of suffering. Check the appearance of the mitochondrial network, and check, of course, the possible contamination with bacteria, mold, or yeast. Okay, so finally, what I would say, it is just that it is crucial when performing live cell microscopy that cell viability is at the forefront, really a forefront of any measurement, to ensure that the physiological and biological processes under investigation are not altered in any way. It is not easy, it will take a lot of work, and it is just the starting point of your experiment. Okay? Thank you.